Okay, fine. Right, can I just check? Have I started sharing the screen, or can you see anything? You can't. Guys on Teams. I don't know if I'm sharing or not.
I can only see you at the moment.
Yeah, okay, let me try again. That one. Okay, can you see now?
Yeah.
Yeah, okay, cool. All right. So, guys, this diagram, which we had a go describing before the break, this contributes to question 4 in this assignment too. So make sure you keep it safe and you type it up, and then you can use it as a basis for the answer to that question. Okay. All right. So meanwhile, I'm going to just carry on with the next slide. So for the other components of blood, I'm kind of giving you a very brief overview, but I'm not going into a lot of detail. So for the purposes of assignment, it's probably going to be more of a question of you writing a short paragraph per each of the components of blood. In the assignment brief, majority of this information goes into a table. So just to sum up some of the key points about white blood cells, I think we already discussed that they are your body's way of protecting itself from bacteria, viruses, foreign proteins, things like that. So essentially they're forming really important part of your immune system. Now you can clearly see from the diagram, from this, not really a diagram, it's called microgram, because this is like a photo of a view which you would be able to see under microscope. You can clearly see that they are larger than red blood cells. They are not as numerous, and also they do contain nucleus, right? So unlike red blood cells, they do have nuclei. So these are the sort of the average number per milliliter, or that's the same thing, so essentially, right? Per milliliter of blood. And there are two main types of white blood cells. Now, you're going to be discussing types of white blood cells and what they do in a lot more detail in your infectious diseases assignment, which you're doing, or not even assignment, unit, which you're doing with Julie. I don't think you've started it yet. The source is probably be at some point after Christmas. So then you're going to be talking about white blood cells in more detail. So for this assignment, it's more of a summary rather than detailed description of types of white blood cells. So it would be nice if you include at least sort of very brief overview of these two types. So phagocytes is the first type, lymphocytes is the second type. Now, so again, I'm not gonna I'm a bit reluctant to kind of stop here and talk about it too long. This is just some of the terminology associated with these two types of white blood cells. Sometimes phagocytes are also known in literature as granulocytes because they have sort of granular appearance of cytoplasm under microscopes. And lymphocytes are agranulocytes. So basically it means without grains within the cytoplasm, so they have clear cytoplasm. So somebody basically looked at the microscope and decided what shall we call them, and they came up with these two terms just to distinguish between these two types of white blood cells, and based on their appearance. Right, so just in a nutshell, just to give you a very, as I said, brief overview, phagocytes help you to get rid of bacteria and viruses through a process called phagocytosis. Does that sound familiar in any shape or form? Where did you hear about phagocytosis before? When did you come across it? In
School, I think.
Okay, in school, and also, did you have to talk about it in cell biology assignment by any chance? Does anybody remember that? Cell biology? No, already forgotten.
What's that? Phagocytosis.
Phagocytosis, yeah? Yeah, actually, one of Rebecca's remembers that it was in cell biology. Can somebody tell me what was happening in phagocytosis? So the same question to teams, yeah? Go on.
Engulfs a solid particle.
Right, so you should quickly found your notes and so much. So it's basically the way phagocytes, you're absolutely right, engulf particles. So here I've got like a little mini animation which shows that the phagocyte, cell of a phagocyte, and the green bits are bacteria. And essentially what happens is that those phagocytes end up kind of engulfing, well, swallowing in inverted commas, that bacteria. And then the bacteria gets destroyed by using digestive enzymes inside lysosomes of those phagocytes. I did have a video, let me see if it's any good. Guys, on those of you on Teams, can you let me know if you can hear the sound? Remind me, let's go away. Is that gonna work? No, thanks. Okay, let me try again. No. Let me try it a different way. How about that? Paste. Go away. Right, so here you can you see that thingy, right? So these little shapes are bacteria, and those bigger blobby shapes are phagocytes. So you can see it kind of just wrapping around this bunch of bacteria until the bacteria end up inside the phagocytes, and that's what's happening. For example, if you have a bit of infection, right? So then that phagocyte is gonna digest that bacteria. It could be a group of viruses, for example. It could, right? So there's another picture here, so you can see how it sort of oozes around them, and that group of bacteria viruses are going to end up inside phagocytes until it gets digested. So it's just a little video to show you the idea behind it. Okay. Right, so phagocytes, they are so-called non-specific, provide non-specific response. What that means is that phagocytes, if you like, don't care, again, in inverted commas, what kind of invader they're coming across. So whatever it might be, they are just going to act on it and engulf it, regardless of what type of infection it is. So that's why it's non-specific. So they're a little bit like kind of equivalent to a community police officer, which is just walking around the streets and they notice there is trouble somewhere and they go and investigate and see what happens. Whereas lymphocytes, they are a lot more sophisticated. What's going on? What's going on? Yeah, is it that one? Yeah. Okay, let's switch it off. Whereas lymphocytes are more, as I said, more sophisticated, so they are providing specific, so-called specific immune response. So they're able to essentially recognize a particular virus, a particular bacteria, and deal with it in that particular way. And that's what the kind of the specific immune response is what provides you with immunity. So if your body was already infected once with one particular pathogen, then lymphocytes are able to recognize the same pathogen again and deal with it more effectively. And that's what vaccines are using, that quality of lymphocytes to, if you like, memorize which pathogen they encountered in the past and keep that memory so that next time they're a lot more efficient in dealing with that particular pathogen. So they're a little bit like MI5 kind of, CIA type guys, who are a lot more know exactly which kind of pathogen they're dealing with, or at least their job is to recognize particular pathogens. Whereas phagocytes, as I said, they're more kind of just a blanket approach. Anything which is foreign, I'm going to just come across it and engulf it.
What is it
The more you
called that
get
allows
sick, the better. Sorry.
No, it's all right.
Was that?
Oh,
Go on,
I was
Kasia.
going to say the more you get sick, the better, really. Then.
Well, it's not necessarily the case. It's just if you encountered a particular pathogen and your body was able to deal with it successfully, it's still going to contain memory for a period of time. Well, those cells which are produced, they're actually called memory cells, right? So you could sort of use that as a term potentially, right? So your body is able to recognize the same pathogen again, and that's what that property of lymphocytes are used in vaccinations, because you essentially introduce tiny bit of this pathogen, lymphocytes learn to recognize it, so next time the same pathogen invades your body, they're a lot better at recognizing it again and dealing with much quicker, right? And that's what the whole kind of idea of vaccination is based around. Okay. So I'm not going to talk about it in more detail here, right? Because for purposes of assignment, you need to sort of more of a summary than in detail information. But if you're interested in this topic, if you scroll towards the end of the PowerPoint, there is a few slides with extra information which gives you some a bit more information on types of lymphocytes and more specifically later. Okay. Right. Any questions from teams at this point? Okay. All right. Oh, I had one question actually for all of you, which is a bit of an interesting fact. If you have, let's say, a cat, yeah, and then it got a little bit infected, and you get this kind of white stuff, basically, which is what we call pus. Does everybody know what pus is? What is in it, in this stuff, in this white, disgusting, slightly smelly stuff? What is it? Any idea at all?
Is it dead cells like being pushed out?
Dead cells. What kind of cells? You're kind of right, but you mean dead cells like skin cells? Well, let's talk about specifically if it's a skin cut. Let's sort of try to narrow it down. So are you talking about like dead skin cells, or you mean something else?
I mean, so basically like whatever's gotten into it. So if it's like a bacteria, so it's been pushed out.
Dead cells of the pathogens.
Yeah.
Oh, okay, I see. Got it. Gotcha. Right. So you again sort of. Oh, okay. So we have another a couple more suggestions on teams saying white blood cells. So you're all not far off. So that white stuff, that pus, what it actually is are essentially phagocyte cells which engulf so many bacteria that they couldn't cope with it anymore and they died. So it's basically the bodies of the warriors which put their life down on your behalf to free you from infection, right? So next time we see, which is slightly gross, but it kind of got this like really interesting story behind it. So it's all these phagocytes or white blood cells which essentially consumed a lot of that bacteria, so to the point where they just are unable to cope anymore, and so you have this kind of mass of dead phagocytes, which becomes pus. Okay, so it's quite an interesting thing. So this phagocytosis was just another random fact. It was discovered by a Russian guy, actually, Mechnikov, and he got Nobel Prize for his discovery of phagocytosis.
When was it discovered?
Oh, good question. You know what? I'm sort of. I think I'm going to probably give you wrong information, but it's sort of like a. Without googling it, maybe somebody can Google it. I'd say like 1930s, maybe
Really?
40s, something like that. I might be wrong, but have a look. But usually, if you just Google when Nobel Prize was awarded, it probably is not going to be the year, because it always takes a long time for them to acknowledge the discovery. So if you're looking this up, you need to be looking at when he was actually working on this topic, rather than when the Nobel Prize was awarded to him. Anyway, so that was a little overview of a lymphocyte.
1869.
Oh, 1869. Bloody hell.
Yeah.
Right, so even earlier than.
Discovered a substance containing both phosphorus and nitrogen in the nuclei of white blood cells found in pus.
Right. Okay. Okay. So even earlier than I thought in that case. Right, and so the last kind of little point I wanted to make is about platelets. So we already kind of just talked about it briefly. Let me again maybe increase the size of this slide. Okay, there we go. So in essence, when you get a cut, these platelets produce particular type of protein called fibrin, and this fibrin looks a little bit like a web of fibers. And what happens that those fibers then have red blood cells and other cells get caught in them. And then this dries up to produce a scab, essentially, or it could be a clot if it's internal cut. So when you have bruise, in essence, it's like a scab, but on the inside of your body rather than outside. That's basically the kind of a summary about platelets. Now, the actual process of blood clotting is incredibly complicated. So don't try to go there and start digging into much detail for the assignment, because you're just going to get yourself mightily confused. If we're talking about medical students who are specializing in, say, cardiovascular system and blood properties, they might have a whole unit on just blood clotting, because it's mind-bogglingly complicated. It's really complex biochemistry. Lots of kind of like it's a cascade of chemical reactions which have to take place in order for the blood to clot. I think I've got a little animation again, but I'm just waiting for people to finish writing information down, so just give me a minute.
Do you have to put the size as an assignment, really? You know how you put like 150,000 to 400,000 per millimetre? Yeah. Do you have to put that in your assignment?
Well, it's up to you, yeah? I'd say for the assignment, the same as for respiratory system. What I suggest is, show me that you've done a bit of independent research. That's what I'm looking for higher grades, yeah? So make sure you summarize what we covered in class, plus a few extra facts that maybe weren't, explicitly described. So anything to do with sizes, maybe numbers, some kind of like extra bit of information. All this counts towards higher grade. Okay, so let me see if I can find this. Yeah. I suggest you don't write all of this down. Yeah, you can leave it and come back to it in your own time. But can we just quickly look at this animation? Of course not. It doesn't want to copy, so let me do it the roundabout way. Copy and paste. Let's hope it's gonna work. Can I just check, guys, on Teams, can you actually hear the sound for the video? Yeah. Oh, okay, so it does work. All right, cool.
Even under physiological conditions, a small tear can appear in the wall of a blood vessel. In order to prevent blood loss, platelets and coagulation factors, such as the pivotal factor 10, acting in a coordinated manner, close this wound. The platelets are responsible for a first sealing of the tear. Then, a number of coagulation factors are activated, leading to the formation of fibrin strands which stabilize the growing clot.
Okay, so that's pretty much it, yeah. Yeah. So you can see how the white little star shapes are platelets. They're producing those that mesh of fibrin fibers, which are this yellow zigzag sort of squiggly lines, and that mesh gets red blood cells. It could be white blood cells gets caught in it, and that's what's kind of forming that blood clot. Okay, and after a while, that tear is going to be patched up, new cells are going to be produced, and so there's not going to be danger of you losing a lot of blood for this little bit of damage. Okay, so you need a bunch of these special proteins called clotting factors for that process to work as well. Does anybody know the name of a condition where people do not clot blood? They're unable to. Hemophilia, exactly. So when we get to genetic disorders, we are going to actually come back and talk about hemophilia at that point. Okay. Okay, moving on back to the PowerPoint. Where am I? Okay. I think that's it, isn't it? Oh, yeah. So let's talk about plasma next briefly. So I'm going to go back to the slide. Current slide. So plasma, as discussed earlier, this is liquid part of your blood, and it transports all sorts of substances. For the purposes of assignment, I'm asking you to include some specific examples of these substances. So can we have a think about the first, let's say, 3 bullet points? I want us to spend a couple of minutes just thinking in our heads first, and then again on Teams, if you're going to start posting some ideas to each other. Can you give me some examples of soluble food molecules, so something which you're going to digest down and then that's going to get passed into plasma to be delivered to individual cells? Can you think of 1 or 2 waste products which would be carried in plasma? And can you think of 1 or 2 hormones, so specific examples of hormones which would be carried in plasma? And when it comes to hormones, can you tell me specifically where those hormones are produced and where they are going to be carried by plasma to? Is it all cells or some group of specific cells? So can everybody have a little go at thinking of some specific examples for the first 3 bullet points? Have you started digestion assignment, haven't you? the unit.
Just been told about it. We haven't really gone into it yet.
Okay, but you haven't really kind of gone in detail. Okay, that's fine. So maybe if you can think of like one example per bullet point, whatever comes to mind. Yeah. So if you really can't think of any specific examples, just keep it and do the ones which you can think of. Okay. So a few people having suggestions on Teams. By the way, you guys, even though you're in class, you're more than welcome to log into Teams and keep track of the conversation which is going on Teams if you find that's helpful. Right, so do you think we should start discussing some answers? Okay, guys on Teams, also make sure you unmute yourself if you have suggestions. So for soluble food molecules, what do we have?
I have glucose.
Okay, glucose, good one, and somebody had it on as one of the suggestions. Nikita, well done. On Teams, glucose. So what do you need to digest in order to get to glucose? So what are we eating?
Sugar.
Well, you could be eating sugar, but what else could you be eating?
Carbohydrates?
Carbohydrates, absolutely right. So anything containing carbohydrates, so things like potato, pasta, cereal. So you don't even have to eat cake or suck on a lollipop or anything like that. You can be just eating perfectly healthy diet, that's fine. You know, you still need a source of glucose, but 100%. But you're not going to be having any carbohydrates in your bloodstream, but you will be having glucose. Why can't we put carbohydrates straight into our bloodstream and let cells break it down into glucose? Somebody have an idea why that has to happen?
It probably has other things in it, like fat, maybe, or not.
All the carbohydrates and fats are different substances anyway. Why can't we? And same question to everybody on Teams. Why can't we just take carbohydrate, drag it straight into your bloodstream, and let that circle around your body in the bloodstream?
Because it's too large.
Because it's
Big. Large.
It's large. Exactly. That's the one. Let's try all the words just to see which one works. They're too large in essence, right? So the protein channels, you remember you have to have specific doors in your cells to let substances in and out. Those doors, those protein channels, are not big enough to accommodate big molecules. So you can get glucose in and out of cells, you can't get carbohydrates in and out of cells. So that's why you have to digest them first. Okay, right. So glucose was one example. Anybody else had other suggestions? Right.
Calcium.
Well, yeah, okay, so things like ions, okay? All right. Right, okay, hold on a minute, let me see. Oh, Sarah Jane, you had a suggestion for another soluble food molecule, which I liked. What was it?
Amino acids?
Amino acids, spot on. Now, Steph early on had suggestion proteins as being one of the nutrients. But are we going to eat a chicken, right? And are we going to drag chicken protein straight in our bloodstream? No. We're going to break it down into, Sarah Jane, amino acids. Exactly. So when you talk about soluble food molecules, you're not going to be talking about proteins as such, because you're going to be digesting those proteins into smaller parts. In the same way as you were digesting carbohydrates into glucose, you're digesting proteins you're eating into smaller molecules called amino acids. Yes, so things like, what are they called? Vitamins and ions, like what Jordan had suggestion, calcium, etc. They're going to be micronutrients. Yeah, you're right. Whereas something like amino acids or glucose are going to be macronutrients, so-called macronutrients. Okay, good. So these can be your couple of suggestions for soluble food molecules. What about waste products?
What about dead cells? Does it transport the dead cells?
Well, I mean, obviously it transports things like platelets, essentially fragments of dead cells, right? But I'm more looking for substances here.
CO2.
CO2, absolutely right. Carbon dioxide, well done. Whichever waste product, we don't usually think of it as waste product, but really it is waste product of aerobic respiration. What do you think I'm talking about?
H2O.
H2O, water, absolutely. Any excess water also going to end up in your bloodstream, won't it? And that's why you've got to wee some of it out, some of this excess fluid, okay? Now, you might go and think of other waste products. I think, do you remember we talked about lactic acid as being one of the waste products of anaerobic respiration? If you're comfortable with that kind of concept, you can add that as another example of waste product, even though we kind of only mentioned it in passing in one of the lectures. Okay, with hormones, well, you know, ton of examples you could use. I think somebody had an example, was it Rebecca? You had one.
Are you talking Rebecca Fenner?
Yeah, Rebecca Fenner, I think. I was just
Insulin.
trying to think. Insulin, right, exactly. Because that's something we talked about in regulation control unit. So insulin, you're producing it where, obviously? Hope everybody remembers. Where do you produce insulin? In your pancreas. That's going to go into your bloodstream, into plasma. And where is it going to be transported to? Well, not just liver. You're thinking about glycogen being stored in the liver, but insulin. To the cell, to every cell in your body. Why is that? Because every cell needs to have their glucose channels open to allow glucose go in. Right, can I leave another example of hormone up to you? Okay, so do a bit of your own digging, okay? And final bullet point, I also would like you to research 1 or 2 common proteins found in plasma. So that's not proteins which you got through, you know, directly from food. These are your human proteins, which your body synthesized. One example would be something like albumin, for example, just to put you on the right track. And then I would like you to do a bit of independent research into that. So let me just write it down as one of the examples. Would be something like albumin. So, so can you do a bit of independent research to include into your assignment? Okay, so let me then show you the assignment brief next, and just kind of have a little discussion about what we've covered today and how it relates to the questions in the assignment. So, where are we here? Okay, assignment 2, respiratory and cardio, that's the one I need. Again. So as I said, this assignment is probably the longest out of a lot. So you can see it's got a bunch of learning criteria which need to be covered. So it's in the form of question and answer. So there's no essay type questions, it's just literally question and answer. What I'd like you to do when you're submitting it, you don't need to include the front sheet, because I think it sort of spooks people because of similarity percentage. So you can delete that if you wanted to. But could you please keep the text of the question themselves, so I can keep track of which question you're answering, which question you own. So all you do is just save it as a Word document, start on the next line and start typing your answer to that question 1. Okay, and then obviously you can include diagrams, include references, whatever else you need to include. All references can, by the way, be at the end of the assignment. You don't have to do it references for each per question. You can just have it as a list at the end.
Even for the images?
Even for the images, yeah.
So do we still need to reference it in the text?
Yeah, ideally as a citation, yes. So if you're using a reference on your list, then ideally you also need to have it as a citation.
I just got confused.
Okay. So let's talk about what we've covered today. So question 1, we're leaving until next week. We're going to be talking about blood vessels next week. But what we did cover is this one, role and structure of red blood cells. Please include a diagram. Some people draw their own, but you don't have to. You can certainly include one from online or from a book. Make sure you explain how structure of red blood cells is adapted to perform its function. Okay. So the next bit is a table, as I mentioned earlier, and essentially your job is to write a largish paragraph per item explaining the function of each of these components. And make sure, can you see here, I'm looking for specific examples of nutrients, gases, waste products, proteins found in plasma. So as I said, proteins would be something like albumin, for example, as we discussed earlier. Okay. If you feel that you want to still include a diagram, you can certainly do that in this section. So for example, for white blood cell of platelets.
Gases, you see CO2 and...
Yeah, so gases will be CO2 and oxygen are the 2 main gases. You do have some nitrogen as well dissolved in plasma, but I'll let you kind of look at that as we saw. All right. So then the next bit, can you see here, explain the transport of oxygen and carbon dioxide in blood. We have not done carbon dioxide yet, but we have talked today about transport of oxygen. So that slide, which we saw earlier, we asked you to describe it in detail. Essentially, that's part of that question. Okay? All right. So in essence, you already have beginning of that question done today.
So basically what we've done is
question 2, question 3, and part of question 4. Okay. Oh, I've done something funny here. Okay. So my suggestion, as I mentioned at the beginning of the session, is try to complete those questions as we're going along, rather than leave them, forget about them, and you realize you have this massive assignment looming. Okay. So try and maybe do it this weekend, or even tonight. You have a couple of hours to sit down, write it all up, do a bit of extra research, and that's done. Okay. So that's where we are. So next week we are looking at transport of carbon dioxide, that bit, and we are looking at the blood vessels. Okay. So that's the aim for next week. Right. So what are we doing next? Next, we are going to do a little session. Let me just see if there is no any comments or anything. I haven't responded to. Okay. Okay, so the next bit is, if you were diligent and you did some summer activities, you might recognize that. Does anybody do it in summer? Maybe not. Nah.
Exercise.
Yeah, well, no. Okay. So can I do a little bit of a sort of overview of human blood groups? Can you see I'm saying here, it's not explicitly in the criteria in the assignment 2, but because majority of you are going into health professions, I think it's super useful bit of information to know about human blood groups. I hope you can appreciate that. So that's why I'm spending a little bit of time covering this particular topic. So, a little bit of overview about blood groups. So there are two main ways of classifying human blood, two main kind of systems. The first system is called ABO, so that's not zero, that's an O, so ABO system. And then the second system is known as rhesus factor, and it's usually written as capital R, little h. So RH of rhesus factor. Really should be little h. Let me just change it like this. I see, this is a funny one, funny. It's font, that's why it doesn't let me do it. Why doesn't it do it? Annoying. Anyway, it's supposed to be capital R, little h. It's written like here, that's better. It's only because it's using funny font. So what are all these blood groups about? Well, they're essentially about presence or absence of particular type of proteins on the surface of your red blood cells. So that applies only, those proteins are only found on red blood cells, not white blood cells. And these proteins are known as antigens. Can you see this word here? Okay. And basically, the idea is that these proteins all have unique shapes. So if you have a particular protein of a particular shape, so somebody decided we're going to call that A antigen, and somebody who has got red blood cells which have got those particular types of proteins, they are known as having blood type A or blood group A.
So it's based on the shape of the blood.
So it's basically not based on the shape of red blood cells, but it's based on the shape of those proteins which are found in the surface of red blood cells. Now, first of all, these diagrams, they're not super realistic. Why is that? Because A, those proteins, they're not green triangles and blue blue balls. Yeah, that didn't come right. Yeah, so much. But they're actually sort of complex 3D shapes, right? And so these shapes is just representation of those antigens. Secondly, they're absolutely tiny. So if you're looking at the microscope, it's not you can look at the red blood cell and you will see like things sticking out of it. They are, we're talking about on like really, really tiny scale here. So they're obviously exaggerated here so you can sort of get the logic of it. So anyway, so then if you have a different shape of protein, so this is known as and then somebody who has got that particular type of protein on their surface of red blood cells are known as having blood group B. Okay. And then you could also have situation where somebody's got red blood cells which contain both types of antigens, so both shapes of proteins, and they're then known as having blood group AB. And then the final scenario is where you have red blood cells with no proteins, no antigens at all, and they're known as blood group O. So this is not zero, by the way. Some people look at it and think it's blood group zero. It's blood group O. Okay, so no antigens. So that's the idea about the four blood groups. Okay. Why is that important to know? Because if you try to, for example, transfuse blood group B into somebody who's got blood group A, the immune system of the person with blood group A is gonna come across these foreign antigens and say, I don't know what you are, you must be invader. You must be a bacteria or a virus or a nasty thing, which I need to get rid of, I need to attack. So imagine a situation, you've got somebody, you've got a patient who's already suffered from blood loss, they're already weakened, and then you are kind of putting into that blood, which their body recognizes foreign, and they start attacking, immune system start to attack those red blood cells. So you have this kind of double whammy. So somebody was already weakened through blood loss, plus they have the immune response flaring up, and that could easily lead to death.
So when you have a blood transfusion and you get your blood type, you can only have your O or O.
Right, well, so that's what I would like you to figure out next, okay? So keeping that in mind, that your immune system of that person is going to be sort of comparing, if you like, the shapes of the antigens and saying, am I familiar with that protein? And if the answer is no, then that blood is going to get rejected. So I'd like you to have a look at this table. So no cheating. No looking it up online, okay? I'm relying particular people on Teams, right? Because it's easy for you to just quickly look it up. So don't be tempted. Do not consult the web. Can you quickly do like a sketch of this table, yeah, in your notepad? And can you, for me, put in a bunch of either ticks, crosses, depending whether that particular combination of blood donor and recipient is allowed, it's good to go, we're fine, we're safe, or whether it's not a good idea. Okay. So I'm going to give you all a few minutes to mull it over. So no looking it up in books or online, just from what you heard just now. Try to see if you can figure it out. Again, guys on Teams, by all means talk to each other, and the same goes for you guys here. If you want to start off on your own, maybe do as many as you can, and then start maybe discussing the ones you're not 100% sure about. So it's just a bit of sort of common sense and a bit of logic. I'm not asking you to know anything in advance, okay? Or use some kind of knowledge from GCSEs or something you remember. It's just literally purely from what we discussed just now. Okay, so we're doing this task right now, okay? Just to make it clear. Right, all done here at this end. Yeah, okay, yeah. Right, so let me just check. Guys on Teams, do you need more time? Or not? Right, so done. Okay, cool. Right, so can we start sort of checking the answers? So if you are donor with a no and recipient is with a O,
Yeah.
yes, of course, yeah, good. So tick here. Donor A, recipient O.
Yes.
Yes. Yes.
Yeah. No.
Yes.
No.
No. Okay, we already have disagreement. So if anybody wants on Teams to chip in as well. So if you have donor with blood group A and recipient with a blood group O, am I allowed to do that?
No.
Why not?
Don't know.
Don't know.
It's got the antibodies on which O doesn't have, so it will think it's a...
So let's think about it, right? So we've got somebody who is blood group O, right? No antigens. And we are putting blood group A into them. Would the immune system be able to recognize this antigen A? No, because they're saying, the immune system is saying, never seen it before, have no idea what it is, must be a nasty.
I was thinking the other way around.
Yeah, let's get rid of,
everyone
let's
can have
attack it.
overall.
Yeah.
That's what I was looking at.
So it's about sort of thinking about it in a logical way. Okay. So let's go back to this next slide then. So that's going to be a no. Okay. And then if you have B and the recipient O, no. AB, no, right? So the immune system of that recipient is not able to deal with any other blood type.
It can give to all.
Okay. So we'll come to that, yeah? So then if your blood donor O, can you give to A?
Yes.
Yes, absolutely right. Yeah, so let's go quickly to here. So if you are with that blood type and I'm putting blood group O, so there's nothing, there is no antigens to recognize. So their immune system is going to be, yeah, fine. Yeah, no problem. They seem like good cells. Yeah, please come in. Yeah, no trouble at all. Right, so we're all good here. A and A.
Yeah.
Yes. What about B and A?
No.
No. What about A, B, and A? A lot of people get confused here.
I put yeah, but I don't think it is. I think it's no.
Right, so we need a bit of help again from Teams. What are we saying? Yes, yay or nay? This one. A, B donor, recipient A.
I have put it be no because it's also got the B there.
I think yeah.
I put yes because it's still got A.
I think
It's
yeah.
not going to fight its own, is it?
Surely.
Yeah, but it would fight.
Would it be like the last resort? That would.
Right, so we have a bit of disagreement here. Let's again try to think about it logically. So we've got, let me just check. So we've got A, isn't it? So we've got this person, and we are going to introduce those sort of cells to them. Okay, so what's the immune system going to think?
It's going to accept the A antigen,
But what about B?
Attack it.
Attack it, and therefore we're going to attack that whole cell anyway. So even though they are recognizing A, they're going to say, yeah, that's okay, but I don't know what this thing is, excuse me, and therefore I'm going to attack that whole cell, right? So it's still a no. Okay, so it's a cross here, I'm
Would they
afraid.
see it as a virus, like infection?
Essentially, anything which that immune system is not able to recognize as your own cell, something which is safe, is going to
Don't
attack.
viruses infect cells?
Yeah, but
So
but
would it
also
be
it can be
like-
recognized by the system which triggers recognition mechanism
Right, is
of
that
white
basically
blood
what that
cells.
is?
Yeah. So and it's all essentially based on lock and key, right? It's all about shapes. Do I recognize this shape? Yeah, you're good guy. Don't know what you are, you're bad guy.
So
right?
basically, we can only really recognise its own or
Or the same as, yeah, the same type of antigen as you already have. Okay, anyway, next one. Blood donor O and recipient B, yes or no? Yes.
Yeah.
A and B? No. B and B?
Yeah.
A, B and B?
No.
No. Okay, O and AB.
Yeah.
Next one.
Yeah.
Yes. Now, a lot of people get confused because, oh, hold on a minute, how come I wasn't able to put AB into A, but I'm able to do the opposite? Well, let's think about it again. So we've got this person this time, and we're putting that blood into them. Would this person's immune system be able to recognize A gene?
Yes.
Yes, absolutely. We're going to say, yeah, good, yeah, no problem. I know what you are. I've seen that before. So therefore, it's going to be a yes. Okay. B,
Yeah.
yes, and finally B,
Yeah.
yes. Okay. So that's your answer. Okay. Hope that makes sense. All right, and then if I just go back for a minute and also mention that, so this is this blood system ABO. There is a second blood system called rhesus factor. And this runs in parallel with ABO. So as well as being one of those blood groups, you also either rhesus positive or RH with a plus. That's what that symbol means, rhesus positive or RH plus. And that means you have yet another type of protein called RH antigen on your surface of the red blood cells. Or if you don't have it, then it's rhesus negative.
Is that why you get like O positive and O negative?
Yes, correct, right? So if you know your blood groups, does anybody know what blood group they are? Your O positive.
Positive.
That'll be the last one.
Huh?
I think I'm O negative.
O negative, yeah. Yeah.
O positive.
So a few people, so people saying A negative, O negative, etc., right? So that negative or positive sign essentially refers to rhesus factor.
So when you give birth,
Yeah. Why is it important to know? And I think Jordan, you already sort of saying a little bit. Does anybody know why it's important to know?
mix any other blood, or just you'll die.
But
Well, if you're pregnant, I know if you're pregnant.
Yeah.
And
You have to have an antigen.
no, if you have a child,
Yeah.
with somebody that's like negative, you're positive, there's a chance that you can miscarry, isn't there? Something
Right,
to do with
so-
miscarrying.
When it comes to pregnancy, it's important to know about negative being rhesus negative and rhesus positive. That's the reason, one of the key reasons why they take blood sample when they find out if you're pregnant. They usually straight away send for a blood sample to find out that information. So if you are rhesus positive, you're fine. But if you're rhesus negative, then you might have to have injections at a certain stage of your pregnancy to make sure that there is no immune conflict, because those antibodies from the unborn child of rhesus positive could potentially get transferred into the mom's blood. Okay, and that's why it's important to know that. Okay, good. So let me see. Oh, let me show you this quick video again. I'll have to copy and paste. I'm guessing. So copy and paste. Yes. So this is just a little summary of everything we're talking about.
It's often said that despite humanity's many conflicts, we all bleed the same blood. It's a nice thought, but not quite accurate. In fact, our blood comes in a few different varieties. Our red blood cells contain a protein called hemoglobin that binds to oxygen. allowing the cells to transport it throughout the body. But they also have another kind of complex protein on the outside of the cell membrane. These proteins, known as antigens, communicate with white blood cells, immune cells that protect against infection. Antigens serve as identifying markers, allowing the immune system to recognize your body's own cells without attacking them as foreign bodies. The two main kinds of antigens, A and B, determine your blood type. But how do we get 4 blood types from only 2 antigens? Well, the antigens are coded for by 3 different alleles, varieties of a particular gene. While the A and B alleles code for A and B antigens, the O allele codes for neither. And because we inherit 1 copy of each gene from a parent, every individual has 2 alleles determining blood type. When these happen to be different, one overrides the other, depending on their relative dominance. For blood types, the A and B alleles are both dominant, while O is recessive. So A and A gives you type A blood, while B and B gives you type B. If you inherit one of each, the resulting codominance will produce both A and B antigens, which is type AB. The O allele is recessive, so either of the others will override it when they're paired, resulting in either type A or type B. But if you happen to inherit 2 O's, instructions will be expressed that make blood cells without the A or the B antigen. Because of these interactions, knowing both parents' blood types lets us predict the relative probability of their children's blood types. Why do blood types matter? For blood transfusions, finding the correct one is a matter of life and death. If someone with type A blood is given type B blood, or vice versa, their antibodies will reject the foreign antigens and attack them, potentially causing the transfused blood to clot. But because people with type AB blood produce both A and B antigens, they don't make antibodies against them, so they will recognize either as safe, making them universal recipients. On the other hand, people with blood type O do not produce either antigen, which makes them universal donors, but will cause their immune system to make antibodies that any other blood type. Unfortunately, matching donors and recipients is a bit more complicated due to additional antigen systems, particularly Rh factor, named after the rhesus monkeys in which it was first isolated. Rh plus or Rh minus refers to the presence or absence of the D antigen of the Rh blood group system. And in addition to impeding some blood transfusions, it can cause severe complications in pregnancy. If an Rh negative mother is carrying an Rh positive child, her body will produce Rh antibodies that may cross the placenta and attack the fetus, a condition known as hemolytic disease of the newborn. Some cultures believe blood type to be associated with personality, though this is not supported by science. And though the proportions of different blood types vary between human populations, scientists aren't sure why they evolved, perhaps as protection against blood-borne diseases, or due to random genetic drift. Finally, different species have different sets of antigens. In fact, the 4 main blood types shared by us apes seem paltry in comparison to the 13 types found in dogs.
So, it's rhesus positive and rhesus negative. Is one more dominant than the other then?
Yes, so rhesus positive is dominant and rhesus negative is recessive. So you're more likely to be positive rather than negative.
So if you're a negative and you have a child that's positive.
Yeah, that's what we can cause the issue.
Yeah.
Correct, yeah.
So maybe we bring but we can save lives. If everybody else didn't, we can save lives. Anyone can allow blood. Yeah. I'd give mine, but I'd pass out, I think I will know. I'm a leader.
Right, okay. I was just communicating through Teams. Just... How do I come out of here?
I said that, but then I go.
Da-da-da-da.
Great.
Well,
Oh, I have my TV. I told you.
why is it gone weird? Help somebody.
That's escape.
Yeah, I think that's what I have to do. To know why it's in the go. Right, don't want another thing. Thank you very much. Right, okay. So, can I just something occurred to me actually? Can we go back momentarily to hemoglobin actually? Because when this slide, this video was playing, I sort of suddenly thought, oh, we haven't really talked about it. Where we've got this information about each heme group containing an atom of iron, right? So can you tell me what's going to happen if you don't get enough iron with your diet? If you sort of start thinking about it logically, so you get anemia, and therefore what?
You have no energy.
Yeah, but why?
You can't carry as much oxygen
Why?
because there's not enough iron atoms
Okay, and therefore?
to bond to the oxygen.
Right, and therefore are you able to produce enough hemoglobin if you haven't got enough iron atoms?
No.
No, right, exactly. So if you think about it in a logical way, if you haven't got enough iron atoms, which are coming through your diet, means you won't be able to put together enough hemoglobin molecules, because it's a bit like building a building, right? If you haven't got, I don't know, the foundation, if you haven't got specific building material, you can't put the whole building together. And if you're not carrying enough hemoglobin, what does that mean?
Not carrying as much
Oxygen. And if you're not carrying enough oxygen, that means right. Exactly. So then you're not able to produce enough energy for aerobic respiration, and that's why you're going to then have all these symptoms such as being tired, fatigued, breathless, etc., etc. Right? So all of it is sort of, can you see? So from this one fact, actually, if you think about all the stuff we talked about earlier, you can arrive all the way to the symptoms of anemia. Okay. So just felt something which is worth mentioning. Okay. There is a game which is actually a pretty good blood typing game where you kind of are given various patients and you have to kind of give them the right blood group. But it is, let me see if it works on this computer, because it's using Adobe, and Adobe apparently is going to get discontinued at the end of this year. They're not supporting it anymore. So all these educational games, you see it's working on my computer. On my computer at home, it didn't work. So anyway, I'm not going to do it now, but please, if you like, give it a go at home and have a little play with it. So it's a blood typing game. So basically, you've got this sort of bit of information, and then you're given all these patients, and
so you want us to mention anything about? I know you said not really, but
mention what? Mention what?
Like blood groups.
Blood groups. It's not, as I said, it's not explicitly part of criteria. I won't be able to give you extra grade or remove a grade through you talking about it. But as I said, I think just because of the nature of profession, in a way, I wish it was part of the criteria, because I think it would be logical thing to include. So it's I'll leave that up to you to decide. If you want to include it, by all means include it. But as I said, directly it's not contributing to criteria. Okay. Right, so just to finish things off, I wanted to play another couple of quick videos. And then this game, it's up to you if you want to have a little go at it at home. Yeah, yeah. We're going to finish anyway in like literally 5 minutes. Okay, if you're bursting, go. It's going to be now on the video for posterity. Okay, let me just see. So this is just a quick video of cells flowing through a red blood cells flowing through capillaries.
Let's go through capillaries.
Well, this one, that was a bigger blood vessel, so that would have been a bigger blood vessel. Where you've got blood flowing pretty quickly, that would mean it's a bigger blood vessel. Where it's going fairly slowly, it's indication that it would be in a capillary. So here's clearly a big blood vessel.
Do you know what it reminds me of? You know that scene in Lion King where Mufasa dies and all the balls of them. Reminds me of that.
How much pressure does the heart create?
How much pressure?
Pressure, yeah. Like
Well, you can measure it, obviously.
It's
You know?
systolic and diastolic
Yeah, you
pressure.
can measure it with 120.
Yeah. It's
Well,
about
I think it's
120 over 90, I think.
Concentrate over 90 what?
Yeah. Systolic and diastolic pressure. So it's the amount that comes out compared to coming in.
Well,
What's that
it's it's in
GSI.
weird units, which is sorry, I've got sort of mental block. Let me think about it. I'll think about it in a minute.
Heart pressure in PSI.
And then another little video about transport of oxygen.
Much as car time.
What time?
You breathe in about 17,000 times per day. It's a process you rarely think about, but behind the scenes, a huge coordinated effort is playing out. Your vital organs, the gut, brain, bones, lungs, blood, and heart, work together to sustain your life by delivering oxygen to tissues throughout your body. Most of our cells need oxygen because it's one of the key ingredients of aerobic respiration. That's the process that produces a molecule called ATP, which our cells use to power their many incredible functions. But getting oxygen throughout our bodies is a surprisingly difficult task. Gas enters cells by diffusing in from their surroundings, and that only happens efficiently over tiny distances. So, for oxygen to reach the cells within our bodies, it needs a transportation network. This is where our 20 trillion red blood cells come in. Each one contains about 270 million oxygen-binding molecules of hemoglobin, which is what gives blood its scarlet hue. To make these cells, the body uses raw materials that become available from the food we eat. So in some ways, you could say that oxygen's journey through the body really begins in the gut. Here, in an amazing display of mechanical and chemical digestion, food gets broken down into its smallest elements, like iron, the building block of hemoglobin. Iron is carried through the cardiovascular system to the body's hematopoietic tissue. This tissue is the birthplace of red blood cells, and it can be found enclosed within our bone marrow cavities. The kidneys regulate our levels of red blood cells through the release of erythropoietin, a hormone which causes marrow to increase production. Our bodies churn out roughly 2.5 million red blood cells per second, a number equivalent to the entire population of Paris, so that oxygen that makes it to the lungs will have ample transportation. But before oxygen can even reach the lungs, the brain needs to get involved. The brainstem initiates breathing by sending a message through your nervous system, all the way to muscles of the diaphragm and ribs. This causes them to contract, thus increasing the space inside the rib cage, which allows the lungs to expand. That expansion drops your lungs' internal air pressure, making air rush in. It's tempting to think of our lungs as two big balloons, but they're actually a lot more complicated than that. Here's why. The red blood cells in the vessels within your lungs can only pick up oxygen molecules that are very close to them. If our lungs were shaped like balloons, air that was not in direct contact with the balloon's inner surface couldn't diffuse through. Luckily, our lungs' architecture ensures that very little oxygen is wasted. Their interior is divided into hundreds of millions of miniature balloon-like projections called alveoli that dramatically increase the contact area to somewhere around 100 square meters. The alveolar walls are made of extremely thin, flat cells that are surrounded by capillaries. Together, the alveolar wall and capillaries make a 2-cell thick membrane that brings blood and oxygen close enough for diffusion. These oxygen-enriched cells are then carried from the lungs through the cardiovascular network, a massive collection of blood vessels that reaches every cell in the body. If we laid this system out end to end in a straight line, the vessels would wrap around the Earth several times. Propelling red blood cells through this extensive network requires a pretty powerful pump, and that's where your heart comes in. The human heart pumps an average of about 100,000 times per day, and it's the powerhouse that ultimately gets oxygen where it needs to go, completing the body's team effort. Just think, this entire complex system is built around the delivery of tiny molecules of oxygen. If just one part malfunctioned, so would we. Breathe in. Your gut, brain, bones, lungs, blood, and heart are continuing their incredible act of coordination that keeps you alive. Breathe out.
Right. Okay. So that's that kind of this morning session. Any last minute questions from teams?
It's 2 PSI. 2 and a bit
No.
PSI.
Oh, my car's got 2.
Okay. So this afternoon we're starting. When are we starting again? 1:15, right? So 1:15 we are looking at. We start looking at Harvard referencing.
Wait,
Are you listening?
tire pressure.
Harvard referencing this afternoon.
Yay!
Okay, so I'll see you all later.
Do you have 2 settings on it? It's got 2 settings on it.
Mine doesn't.
No, when you fill it up.
What are you talking about?